In this video, we're going to be doing mostly work with prepared slides on the microscope, and our first focus will be just on learning to identify xylem and phloem in a cross-section of a stem of a typical non-monocot flowering plant. We'll do medicago, which is alfalfa. Let's go ahead and get a slide on the scope, and we could do our talking around the image. I'm going to start by zooming in right to the important things, xylem and phloem. The first thing that should jump out at you is the xylem. In a typical prepared slide like this, the xylem cells are super easy to identify because they stain red or pink and they have thick walls. Part of the staining procedure includes a red stain that adheres selectively to lignin, which is the brittle waxy material that reinforces the cell walls of sturdy xylem tubes, making them rigid and able to withstand pressure and support weight, of course. And it's also what makes them largely water impermeable because they need to conduct the water without it leaking out. Now, xylem cells only really exist when the xylem is forming. The cells with their membranes, cytoplasm, and everything else are there only during the phase in which the cell wall is constructed largely from cellulose and later to be reinforced with lignin. Once the cell walls have formed this reinforced tubular structure, the actual cell that the wall was built around, it dies and is flushed out by the flow of water through the xylem. There's no cell that's there when the xylem sap is flowing, and that's why the xylem transport is considered apoplastic. So what you're seeing here on the lower left is more mature xylem with fully pink and therefore lignified walls. These are the vessels that would actually be carrying xylem sap up from the roots to the leaves in a living plant. And as you follow xylem up and towards the right, the xylem, is younger and younger. You can see cells with thick walls that are green. These are the ones in which lignin reinforcement has not yet been completed. And then further up, you can see cells that are xylemish in size, but without the thick walls, and within which there's actually cellular stuff evident. These are the youngest xylem, and you can tell they're young, well, because the cells are still in there, and also because this is close to the point where you have the undifferentiated cells that actually make the xylem cells. This is the vascular cambium, a thin layer of cells that's responsible for the production of both xylem and phloem. Cells that it produces toward the inside of the stem differentiate into xylem. They develop larger size, sometimes, not always, and they grow thick walls, and then they die once the xylem walls are adequately complete. Now, the cells that the vascular cambium produces toward the outside of the stem differentiate into phloem, the vascular tissue that carries mostly sugars, redistributing them from where they're abundant to areas where the sugars are needed. So, xylem is actually really easy to identify based on its appearance, red and thick-walled. Phloem cells are identifiable on this medicago stem mostly by their position, toward the outside relative to the xylem. The phloem cells you see here are green and relatively thin-walled. On this medicago slide, you can go past the end of the youngest xylem, right up to around here, because these big cells are very young xylem cells. And at this point, you're in the phloem up until you get up to these green cells with thick walls, which are not phloem. They're basically supporting structures called parenchyma that assist in holding the stem up. They're kind of like the stringy things that you have in a celery stalk. Okay? The phloem is all these green cells in between the parenchyma and the xylem. Yeah, phloem is harder to identify relative to xylem, and you're not always going to have a cap on parenchyma on the outside of the phloem. But, okay, once you find the xylem in a non-monocot stem, you can usually locate the phloem by its position to the outside of the xylem and by the fact that it's usually green. Now, vascular cambium is something that you just have to know is there in between the xylem and the phloem. If anyone ever asks you to approximate the location of the vascular cambium, the thing to do is to locate the xylem and the phloem and estimate where the cells are no longer either xylem or phloem, and right in the middle of that should be the vascular cambium layer. It's easy enough to locate, but what you can't do is identify vascular cambium based on some kind of characteristic appearance. In multicellular organisms, the cells that are recognizable by what they look like are usually the ones that are fully differentiated. They've become the cell that does something in the multicellular organization. Cells that are undifferentiated are generally nondescript and relatively few in number. In animals, we would call undifferentiated populations of cells like this stem cells, while in plants, we refer to such cells as meristem or meristematic tissue. The importance of meristematic tissues like the vascular cambium cannot be overstated. These are the cells responsible for plant growth. In the case of vascular cambium, the need for more and more xylem and phloem in the stem grows as the plant grows in size. More and more xylem, more and more phloem. Mostly xylem. It's all produced in the form of cells splitting away from the vascular cambium and differentiating. It's just one very thin layer of cells, but it's the main center of growth for stems that sometimes grow into the huge trunks of trees. In the last video, I showed you my avocado tree with reed and fuerte avocados, both grafted onto the rootstock of a lousy, tasteless avocado that I bought from Home Depot. The technique of grafting is actually kind of tricky. You have to match up the vascular cambium of both the rootstock and the scion wood in order to get them to fuse together. If you can manage that, then the graft will probably take. But since you can't really see the vascular cambium when you're putting the stems together, you also have to get kind of lucky. In my case, I did about 10 grafts, and out of the 10, only 4 were successful. Medicago, the alfalfa, is an herbaceous annual plant. Herbaceous means that it's non-woody, and annual because it grows from seed to complete its full life through flowering and seed production within a single growth season. These plants are not long-lived, and so the stem of the plant needs only to be sturdy enough to keep the plant upright for a few months at most. Basically, the stem is just a temporary structure. And as you would expect in temporary structures, you don't see a huge investment of resources. It's as sturdy as it needs to be, but just barely. If it had been made less sturdy, the plant's fitness would suffer because its stem would probably have collapsed. And if the plant had made the stems extra sturdy, more than what was necessary, that is, the plant would be wasting resources for stem construction that would be more productively put elsewhere, like in flower and fruit production, something directly related to reproduction. A plant that goes overboard in stem sturdiness would have fewer offspring than a plant that doesn't over-engineer. And this is how natural selection leads plants to evolve stems that are just as sturdy as they need to be. So you look at the Medicago stem and you see a big airspace in the middle. Airspace requires no investment of material, and this makes perfect sense for an herbaceous annual. A woody stem is different because its main bulk, most of the cross-sectional area from the vascular cambium all the way to the center of the stem, will be xylem. Remember that in Greek, xylo means wood. Woody stems are nearly all xylem. We'll address woody stems and the structure of wood in the next video. Right now, let's take a quick look at a monocot stem because they are really different. Much of what I just told you holds true for the majority of non-monocot vascular plants, that there's a vascular cambium. That's the main source of plant growth by continually adding new xylem and new phloem layer by layer. This is called secondary growth, but it's a phenomenon that is lacking in monocots. We'll look at the grass, Zea mays. It's actually corn, which is a grass, as an example of a monocot stem. But this is the same basic structure that you would see in any other monocot. Orchids, lilies, even big palm trees. If you look at the cross section, you see the bundles of vascular tissue are scattered throughout the cross-sectional area. It's not all around the outside of a hollow stem like what we saw in Medicago. The vascular bundles of monocots are also pretty characteristic. Most people see little faces. The eyes, the nose, and the mouth on these faces are all xylem. And you can tell they've got thick, red staining walls, at least when they're mature. The forehead of this face is a cluster of green cells, and these are the phloem. And there's no vascular cambium at all. No additional xylem or phloem that's ever added. The cells in each of these monocot vascular bundles are all fully differentiated. As a plant grows, its need for more xylem and phloem are met by adding additional vascular bundles rather than adding more xylem and more phloem with meristematic tissue that's already there in the stem. Okay, Medicago stem, typical of a non-monocot. Medicago is actually a eudicot. It's an herbaceous stem. Okay, check. Zea mays, also herbaceous, typical of a monocot. Check. Now, on to roots. We'll just look at one kind, the ranunculus, which is a buttercup, also a eudicot. Okay. Now, if you've ever been to the flower fields on Palomar Airport Road off the 5, all of those plants are ranunculus. And if you haven't been there, you've probably seen it. And so I'm going to say that you're already kind of familiar with this plant. Now, there's no particular reason why I'm choosing ranunculus, apart from the fact that it's the plant whose roots seem to be favored by the people who make these prepared slides that we're looking at. It could actually be any plant. Well, any eudicot anyways. Monocots are a little different again. And to be honest, I'm really not sure about the basal flowering plants or gymnosperms. This is one of those situations where our hands are kind of tied by the slides that are widely available for teaching purposes. It would be a real bummer if it turned out that ranunculus roots are unique and most plants' roots are totally different. But I doubt that's the case, because when I look at this root, I see the same structure that I see when I cut carrots. Skin on the outside, a thick layer of spongy, non-fibrous material, and an inner core that does have a more fibrous texture. So I figure, true for a carrot, true for ranunculus. This at least seems to be consistent with my worldview on roots, even if my only experience is with carrots. Now, the skin of the root is a single layer of cells. That's the epidermis. In the mature root, like what we're seeing here, the epidermis is largely impermeable to water. But in young roots, close to the growing tip, the epidermal cells are actually water permeable, and there are even small gaps in between the cells, which allows water from the soil to enter easily through the cells and in between the cells. The cortex is the outer layer underneath the epidermis and going all the way to the vascular stele or core. In this slide, you can see purple inclusions in the cortex. These are leucoplasts, starch storage for the root. Now, we're going to zoom in on the endodermal layer that you can see going all the way around the vascular stele. I want you to think of the endodermis as an impermeable wall separating the cortex from the vascular tissue inside, the xylem and the phloem. The only way across this endodermis are these cells with thinner walls that largely conduct water from the cortex into the xylem symplastically, that is, through these cells rather than around them. Once the water gets into the xylem, the water is again apoplastic, outside of any actual living cell. Now, when you look at the vascular steel in the whole, we're on familiar ground again. The xylem is self-evident, obvious because of the thick walls that are staining pink or red. If you see a starfish inside the vascular core ranunculus, all that is xylem, right? Pink, thick-walled cells. The green cells in between the arms of the starfish, but still inside the endodermis, that's all phloem. Now, the last group of cells that don't stand out, mostly because of their state of being undifferentiated, is the pericycle. Pericycle cells in the root both facilitate the movement of materials across the endodermis, and they also contribute to root growth, both secondary growth with more xylem and more phloem, as well as lateral branching, the production of side branches on the root. Okay, this is the basic root anatomy that we refer to in our lectures on vascular physiology. In those lectures, the root is crucial to our discussion because it's the entry point for water into the xylem, the place where a positive pressure is generated. The xylem in the stems, which we have already seen in Medicago and Zeum, that xylem conducts the water upwards toward the leaves, where the water exits the xylem, creating the negative pressure, and this is what we're going to be looking at next. This time, we'll start with the leaf of Zea mays, the monocot. This is a really nice choice to start with because the parallel venation, characteristic of monocots, right, makes it easy to navigate within the leaf cross-section. In the cornleaf, there's a central vein that's bigger than the multiple smaller veins running parallel. Since this leaf is cut in cross-section, we're looking down at the ends of all the leaf's parallel veins. Okay, so if you look at the central vein, you might see a familiar face, the classic monocot vascular bundle. Okay, now in this leaf, there's a lot that we could be unpacking, but I'm going to ask you only to pay attention to two things on this slide. One is the fact that there are lots of air spaces within the leaf. And two, the fact that there are stomata in the epidermis, the top and the bottom layers of the leaf, that communicate between the air spaces and the outside of the leaf. If we look along the leaf to the side of the central vein, yeah, lots of air spaces. Those are the air spaces where the leaf exchanges CO2 from the air for O2, which is produced as the waste of photosynthesis. Now you can also see that between the airspace and the outside, there are these guard cells that can open or close the space between those thick layers. These guard cells form stomata, or little mouths, that can either open or close. Most of the stomata that you see here are closed, but there are a few that are open like this one. Got it? Okay. Now we're going to look at a different leaf, a non-monocot. We'll use syringa, which is the lilac. If you've ever gone to Julian in the springtime for apple pie, well, first, shame on you. Apple season is in the fall. But in the spring, local growers are usually selling bundles of fragrant purple flowers on woody stems. These are lilacs. And we're looking at the leaves in cross-section. Now you probably want to note that this is clearly different from the Zea leaf. You don't see the ends of lots of vascular bundles because this is not a monocot. Syringa's a eudicot. The vascular bundles are there, but they're going in all different directions, right? The important similarity is the same structures that I had you look for on the Zia. The presence of lots of airspace in the leaf and stomata that communicate the airspace with the outside air. Now, the last thing that I have students do in this lab is to make a wet mount from a little piece of a leaf that they collect outside. You don't even have to do a wet mount. Just get a little piece of a leaf, and here I'm pinching off a bit of a grape leaf, and I'm putting it on the slide and looking at it just the way it is. All you have to do is focus on the surface of the leaf, and there are lots of little mouths, stomata, singular, stoma. These are the plant's openings through which nearly all the CO2 enters the plant as a tiny, tiny fraction of the percent of air. Yes, we're looking at an increase to 420 parts per million sometime really soon. But 420 parts per million is 0.42 parts per thousand, which is 0.042% of the air. And that's a level that hasn't been reached yet, even with the human caused increases in CO2. Okay, despite its rarity, despite CO2's rarity in the air, plants are harvesting the CO2 and using light energy to chemically reduce it to make biomass. While the stomata are open to allow for this uptake of CO2, water is transpiring through those same open stomata from the moist, humid airspace inside the leaf to the relatively dry outside air. This is the water loss that drives xylem movement by creating negative pressure from the top of the xylem column. Okay. I don't know about you, but for me, seeing this stuff in person makes the process of xylem transport so much easier to visualize. I wish I could offer you a more direct personal experience than what you're getting in this video, but for now this will just have to do. Okay. One more video, all about wood.